So I sent you all an email. I've received everybody's field notebook link. It all works. Everything's fine. I haven't started grading them yet. I will, but like I said, it's going to take me a while. But as soon as they're graded, I'm going to be sending you back a PDF of the score sheet, which is, if you look on the instructions, the original instructions, there's a sample of that, and it's by group and then taxonomy and location, and you can maximum amount 3 points in each category and the total. So, and I'll make comments where they are. Sometimes I go through field notebooks and, I don't have anything to comment on. You do fine. Sometimes you write stuff in the insights I like to comment on. Sometimes it just kind of depends on what mood I'm in. So don't feel like if you don't see any comments on something that I don't care about what you put. It may be just a matter of I might be trying to do 2 or 3 other things at once and grade the field. What I like to do is I like to set the field notebooks for a time early in the morning when I don't have to mess with anything else. There's no emails coming in. I can just relax and read them. Because like I told you in class, I actually do enjoy grading these things. Not everybody. Sometimes I'm like, oh my goodness, what a wreck. But I don't expect that to come out in any of y'all. So let's go ahead and get finished up with the arthropods, with the insects, because we still got to move on into the first group of deuterostomes today. You remember, we haven't talked about any deuterostomes at all. We've only talked about, well, way back, the protostomes and deuterostomes. We've only talked about protostomes. There's only 2 deuterostomes out there, 2 phyla of deuterostomes. So let's get to those, but let's finish up the insects. As you can probably imagine, as I've emphasized throughout this entire lecture, there is just a boatload of arthropods out there, not just insects. Insects are only one group of them, but there is a boatload of insects as well. There are these things called the bristles. I'm not an entomologist, so I'm not real familiar with the bristles or Archaeognatha. But from what I do know about them, and looking at the name, mouth parts are very important among many groups of insects. And I guess your mouth parts are always kind of important. But in identifying them is what I mean. In identifying insects, like Dr. Weimer, who works here at school, as an assistant, I don't remember, some title. Anyway, he was an entomologist in college, and he's very, whenever we are looking at insects, he's always checking out the mouth parts and Archaeognatha, archaeo means old, and gnatha means jaw. So Archaeognathids are some of the earliest insects we see in the fossil records. And my guess by the name, it must have something to do with the more underdeveloped nature of their mouths, I guess, the Archaeognatha being the older mouthed type of insects. Like I said, in many cases of insects, especially among the like the Hemiptera right here, it's all about, oh my pen, come on, there we go. Hemiptera, it's all about the mouth parts. Hemipteras have a very distinctive mouth part. It's what makes them hemipterans. Now here, the name, I know I'm getting ahead of myself, I'll come back, but there's just so much to talk about here. The name of the different groups of insects, and there are many orders, and like I said, there's only 6 that are shown here, and I got a couple of more that I threw in, but I'm not going to expect you to know all these orders. But the name in this case, like Hemiptera, literally means semi-winged, semi-hemi, Ptera, P-T-E-R-A, has to do with wings, okay? So what we mean by that is hemipteran insects have, I wrote it there in red, textured and non-textured wings. In other words, they've usually got those outer wings that have, if you can see in the picture of the bug, I got a bigger picture. I know I'm not doing these in order, so I hope you're not one of those folks that bothered. But I do hemipterans. There we go. If you notice, you see right here, these are the outer wings right here, and they'll have a different texture. In many cases, the other wings will be more smooth in texture than the other ones. But the big things about the hemipterans isn't, and they're really uniquely, they're not named because of this, but they have a mouth part that they use for piercing. All right, so if you're thinking about the things like a lot of the insects, they would have piercing mouth parts like stink bugs do, and they can use those to feed on their prey, or they can use that in some cases to sense their environment around them. But that's going to be the big thing about the hemipterans, is going to be that very unique mouth part. And as I said in previous slides, when we're talking about insects, mouth parts are very important because they fill so many different environments, and feed upon so many different things. So let's look at so let's go back ahead. Oh, the Zygentoma, the Zygentomas, the silverfish. I bring those up because this is a neat group, relatively ancient group, a small group, only about 450 species. And it, again, to me, anyway, illustrates how science advances. The changes that have occurred to the Zygenotoma group of arthropods since the time I was in college, because I remember we were discussing them, has been significant. It used to be we thought that all those little silverfish that are crawling around were all members of one order, all in the same order. But now we know that there are different families within that order too. So we're learning, that whole phylogeny thing is changing. We're adding more to our understanding what other members might fit in this group of the Zygenotoma. The winged insects down below, again, the Coleopteras. Coleus is a Latin word that refers to having a sheathed, or like if you had a shield. So like this male snout weevil, in spite of the fact you go, wait a minute, that's got a really distinctive mouth part. You have to have that mouth part that hemipterans do, that small little piercing one. But they have what we call sheathed wings, or they have wings that are covered by an outer covering, which is not a wing. See, the hemipterans have two sets of wings. The Coleopterans have these outer devices that sheathe the wings. The Dipteras are two. So what we're talking about is organisms that only have two wings. In many cases, what's interesting about this, and I'm trying to remember as I'm sitting here why I put the picture of the beetles crossing Abbey Road here. There must have been something when I was thinking about the Diptera. Diptera, the two-winged organisms out there, like these bees, mosquitoes, of course. Mosquitoes, again, with a proboscis mouthpart, but they do belong in the dipterans. What I wanted to say about the dipterans is kind of neat, especially among many species of flies that belong in this group. The dipterans have the other set of wings has been reduced down to balance. They have these little balance things that work. So one set of wings is for flying, and then the other set of wings is what they use for kind of balancing themselves when they fly. The big group in there, 125,000 species, the Hymenoptera, they're the ones known for if you think about bees and wasps, they're very flimsy tissue like a wing. So that's a hymen, is a thin tissue. So that's how you might remember those. The Lepidoptera, they have scaly wings, and if you're looking and they're going, Steve's messed up and he put a doggone bird, let me show you. And there's what I mean by the very thin membranous wings. Okay, there's the Lepidoptera. That's actually a moth. That's not a hummingbird. Isn't that something? That's huge. We have those in Florida. One of my students a few semesters ago actually got a picture of those. Why did I put the picture of the Beatles crossing Abbey Road? See, Paul McCartney's out of step. It must have been something I was thinking when I did that. Goodness. Sometimes I'm going through and I think about when I do a slide, I'm going to add this because it's kind of unique. And then I say, well, I'll remember what I meant by that. And then I've forgotten. But if I remember why I put that, to remember something about Diptera, the way I remember the Diptera, going back for just a second, is Di means two. They only have two wings. See, the other ones have like outer wings and inner wings. Lepidoptera, the butterflies and such, have scaly wings, and lepid literally means scaly. We talked about the hemiptera. We started, the katydids. Look at the katydids. Katydids are Orthoptera, and ortho means straight. So to me, where you get the Orthoptera to remember is the katydids tend to have these long, very prominent, straight parts of their legs, okay? Katydids are very popular insects among kids. Isn't that neat? Look at that critter. See, all modified mouth parts. What do you think these are for, by the way, on the katydid's legs? I'm not sure I know off the top of my head, but you could probably think of something, right? Yeah, that might be some. Yeah, defense. Maybe sensory too. Looking around in their head too. So I'm tending to lean towards what you guys are saying. Probably especially if you think this katydid is obviously in an area. Yeah, that might help too. Sticking around on things, maybe gas exchange too, little tiny. It's probably one of those things that we find about in critters where, can actually do more than one thing. Acceptations, like we've been talking about. Three more groups I did want to look at as far as smart. How sharp? The I'm sorry, the little. They don't look. They may be sharp, but they're probably not real stiff. So I don't know. I mean, if you're a predator of that size, that might be sharp and stiff enough to dissuade you. But I put these last three orders in again because, well, one of them is my favorite, the Odonata. I love dragonflies. I think they're really neat. I think it's probably because they remind me of where we've been, because the dragonflies are so little changed since the time of the dinosaurs. The dragonflies, or the Odonata group of insects. Odonata, by the way, meaning teeth. These guys are the ones with chewing mouth parts. They've been around for a long time. They were probably some of the first insects around, so I think it's kind of cool. It just sort of reminds me how we've come so far, and yet there's still some critters that have just continued to make it. Remember what I said, evolution isn't a race. It isn't a competition. It isn't trying to be the best. It's trying to be the best right now in the environment that you got. And somehow these dragonflies, with very little change through time, have been able to find these niches to allow them to weave their way through all the difference that have occurred on Earth in terms of the environment through time. Neat critters, huh? The Isoptera, iso means equal, so we're talking about, and ptera again means wings. So we're talking about those with equal wings, but if you look, one of the more common isopterans are termites, and they're wingless, except there are certain times when they're, of course, going through the reproductive cycle, they'll have wings, and it would be two pairs of equal-sized wings. Chewing mouth parts. Termites, of course, are well known for the fact that they can digest the wood in your house. They have those symbiotic bacteria that live in their gut like the cows do, that actually help them digest the wood, so they break down the cellulose for them. There are so many termites on Earth, too, that eating cellulose, apparently one of the results of that is you're very flatulent, so you produce a lot of methane. So termites are one of the key contributors to the increase in methane, which is another one of the warming or greenhouse gases, so-called greenhouse gases. But termites are one of the major contributors of methane. Cows are too. Cows are big flatulent critters. But just think how many termites there are to imagine that each little termite pooping every day a little bit, is floating up into the atmosphere and collecting. There must be just a boatload of termites. We all are familiar with fleas, and fleas are very well known for their piercing and sucking mouth parts, which they use, of course, to grab a blood meal. That's what when I always ask students, do we have parasites? And I remind them of the mosquitoes, and I remind them of the fleas and how those are ectoparasites, using those, and they're wingless, okay, with those jumping legs that they use for feeding on blood meals, right? Many times being on mammals, many times being a vector, you can imagine. Now, interesting enough, tell me something about why you would expect maybe organisms like this, these ectoparasitic fleas, why you might expect them to be wingless with big jumping legs. What about that type of lifestyle? How could you put those two together? Say, well, I believe fleas are wingless, or they evolved to be wingless, or they might have actually evolved from a winged ancestor, but why are they wingless? It's kind of the same reason why leeches don't have setae, those little things that help them move around. Well, they don't have a need to fly, do they, Sean? Why don't they really have a need to fly? I mean, the jumping legs helps, I understand, yeah, but what is it about their lifestyle that that would suggest that they're not really critters that move around a lot? Yeah, they kind of hold on to their prey, right? That's something, yeah, there you go. Yes, absolutely. So that's another good observation, Sean. Yeah, you would expect that the critters, since a flea can jump pretty well, but I doubt it can jump, dozens of feet into the air. I don't know. I wonder what the highest. I know fleas can jump several times their body lengths. It'd be like us jumping 60 feet, but yeah, definitely. So that's what I mean about doing that detective work. We say, oh, look at this critter. Now what we know about evolution, we know organisms are adapted to their environment. So what is it about, of being a flea that would suggest that the kind of life it lives would say, well, let's get rid of wings. Remember, things that you don't need are expensive. They sure are. They sure are. Hey, one of the ways to do that, by the way, without introducing a lot of pesticides is to get borax. It's a white powder that you use it sometimes in laundry. You might find it like on the top shelf at Publix or something. But I had a real flea problem. And well, one thing now what I do is we don't have carpet. We just have rugs. So we have tile. But when this house I rented before, when I moved in, had a flea problem, and I put borax down. And I just kind of dusted it into the carpet. I left it. I didn't vacuum it up. But the borax soaked down into the carpet path, and it dries out the flea eggs. So it kind of helped. Of course, you have to control them on the animal as well, too. But yeah, I had this one house I had was terrible. They sense carbon dioxide too. Carbon dioxide gets them all excited. When they sense a high amount of carbon dioxide, they assume there's a lot of breathing critters around in which to get a blood meal. Lovely, lovely animals. Okay, so we're done with all the protostomes. We're done with all the Lophotrochozoa. That's such a neat group, especially when you think about just saying the word, right? You remember why we put these all together is because in many cases they either share a similar type feeding mechanism. We didn't talk about the Ectoprocta, but the Ectoprocta have that lophophore feeding mechanism. And the sort of like, it's sort of like cilia, but they're not cilia. Same sort of they're filter feeders, obviously. And remember the similarity of the larvae, the trochophore larvae. The mollusks had that, as did the annelids. Again, my emphasis on that is being in biology, in evolution, and when we're putting these phylogenetic trees together, similarity of ancestral stages. I think last week I even typed out a sentence there. Ontogeny recapitulates phylogeny. You might think, well, what the heck, right? That basically, if we look at the steps that organisms go through as they develop and grow, the idea we could somehow figure out what their evolutionary lineage was. Now, that's not exactly the case. It more seems that way than it really is. But there is some credence of truth to that, or some credence to scientific justification for that, because organisms that have that start with similar larval stages, or the same exact larval stage, no matter what they end up becoming, that's usually a pretty definite sign that they have a common ancestor. See, because if you think about it, and the reason why I'm going into this is this is important in understanding the group that we're moving into next, the deuterostomes. If you think about it, if you make little tiny changes the way a larvae develop, okay, you can have that can result in very large changes once that larvae grows up. The classic example that we sometimes use, if I showed you the skull of a young chimpanzee infant, and I showed you the skull of a human infant, you probably, unless you were an anthropologist or somebody like that trained, you or we probably couldn't tell the difference between the two. They look pretty identical. Now, an adult chimpanzee skull looks significantly different than a human chimpanzee skull. A human's skull, right? You'd be able to tell the difference between those two, right? The chimpanzee, the forehead is sloped back, the eyes are sunken in, the much more pronounced jaw. I mean, there's no mistaking that, okay? If we look at that and we say, all right, that's an evidence that of similarity of ancestry, and we know humans and we all fit in with the primates, but little tiny changes early in embryonic or larval development can have very big effects. You know, it's kind of like if we were sending you to, let's say, Jupiter, right? And my little slide rule or whatever I used was off in my calculations by 1/100 of an inch. In other words, I had to have you at some point in space to be captured by Jupiter's gravity within 1/100th of an inch. And if you were too far out of that, you were zipped on by Jupiter, and it was so long. And if you were too far to the right of that, you ran into Jupiter or something like that, right? My astrophysics. But anyway, so a 1/10th of an inch, isn't that what I said, or 100th, or whatever that case it was? From here on Earth, that's not a lot. But when you go out to Jupiter, which is many miles, millions of miles away, the distance gets bigger and bigger, doesn't it? I think I talked to you guys early on about one of the strategies for deflecting an asteroid. If we could see them far enough away, far enough out there in space, we could do something like put something in orbit around them that would reflect the sun onto the asteroid and would cause it to start getting hot, like if you were burning up a piece of paper, right? Well, that little hot spot would give off particles and steam, and that would push that asteroid very gently, very like you could probably almost put your force and give it about that much. But if that thing is millions of miles away and you push it gently, by the time it gets to us, it misses us by a long shot, right? So you see what I mean by how we relate that to organisms, right? Little tiny changes in embryonic development can not only, if you look at similarity of embryonic development, you can suggest some ideas about relationship, but those little tiny changes that occur can be, they can be truncated. Mathematicians talk about truncated errors all the time. If you forget to carry the 1 in the very first column, everything else is messed up, right? It's a truncated error. You understand what I mean by that, right? Are we ready now to talk about the deuterostome? You probably think, where in the heck is he going with all this? Well, it's very important in understanding the deuterostomes. All right, the deuterostomes include a group we looked at the other day in class. These were the echinoderms. We'll talk about those first. And then the really big group that I know you all have been waiting all semester long to get to is going to be these guys, the chordates, right? These are the quote-unquote vertebrates, all right? Chordate, the word chordate literally comes from the Latin. It means string, so it refers to our spinal column, okay? Chordates have a spinal column. We talked very early on about that big definition, or that big categorization we learned about animals very early on, right? There were the invertebrates, and then there were the vertebrates. And the vertebrates are in the group with the phylum Chordata. They have a backbone. But as you're going to learn, as you've learned throughout this semester, there are chordates that don't have backbones. All right, so anyway, more on that later on. We're not going to talk about the hemichordates. So they're a neat little group, might be, somewhere kind of in between in the evolutionary. We're more closely related to the echinoderms than we are to any other organisms out there we looked at. Even though we thought at one time maybe it was the annelids, it was that blood vessel and nerve cord switch that kind of threw that one out. The arthropods that was a pretty legitimate hypothesis for a while, though, because, I mean, if you think about it, even morphologically, we kind of are arthropodish, right? We've got appendages that we move and all that sort of stuff. But the way we move our appendages and the way we all evolved and the way we develop is very different. So all of those things that we might see between us and the arthropods would just be an example of that thing that I've by now rammed into your head a million times, right? Why do things sometimes look alike even though they're not related? Please humor me with you. I know you guys know this one. Let me find where I am here so I can see. Yeah, thank you. Remember, that's always a big tripping thing. It's always like, that's always what phylogenists have to be. Emily's just heard something, so she may bark. Oh, they're on the back porch. All righty, so the deuterostomia. Now, why are we in with the deuterostomia? Well, for one thing, because I said, and I just finished saying, similarity of embryonic development, right? Remember back in chapter 33, we talked about that there are protostomes and deuterostomes, and protostomes, the mouth forms first, and then the anus forms second, etc. We share that embryonic path, along with those other types. There was a difference in the way that the cells divided. There was a difference in whether the cells had determinate or indeterminate development. Deuterostome was only part of it, okay? But the fact that we share that similar type of embryonic development with the echinoderms, as I've just been going through with you, says that probably indicates or warrants putting us in a group, the deuterostomia group, indicating that your closest living invertebrate ancestor would be the echinoderms, the sea star. Isn't that weird? For one thing, what's really weird about that is when you look at the echinoderms, so we'll start, okay, so here are the deuterostomes. It's the sharks, the humans, the black rhino. Is that a black? I think it's a black. I can never tell between the black and the white rhino. They're not really black and white. This is a sea cucumber, so that's an echinoderm, and this is an echinoderm that's a Florida echinoderm. So all of these critters, and of course, you know, this is us down here, all of these are lumped together in this group, the Deuterostomia, because like I said, they all have that same sort of embryonic development. Now let's go into the echinoderms and take a look at them, because that's a weird group. It's a weird group to think that that is our closest living ancestor. Hopefully you understand that by me talking to you, but you can understand it by looking at this chart as well. Look. This is when the ancestor of all these protostomes over here, let me see if I can divert Cooper's focus for a second. Cooper, you're done. Come here. Good boy. All right. Just relax. Come here. It's like raising children. Okay, but when you think about it, when you look at the organisms out there, it seems like the echinoderm would be like the less likely group. They look really nothing like us at all, right? Echinodermata. Echinodermata, that word literally means spiny skin. If you ever run your hands over a sea star, it feels that way, doesn't it? It's got a spiny or sort of a rough texture to it. Now, this is an interesting group in many, many ways, but one of the ways that it's interesting is there are no freshwater echinoderms. They're exclusively marine. So this is a group of organisms that evolved in the ocean and just never left. Remember, in many groups, there are some species, well, in many groups, there are those who left, and then in some, there are those who left and then even went back into the ocean. Now, where the weirdness becomes when it comes to understanding or at least accepting that these are our closest ancestors would be, first of all, in symmetry, right? We have bilateral symmetry. We've been talking about bilateral symmetry since the flatworms, right? Flatworms had it. Certainly the arthropods had it. We come to this group here, and we're going back to a type of symmetry, pentaradial. That just means basically radial, but many times with 5. But it's not always 5, okay? The whole idea of having 5 arms is a misnomer. Next slide, right? Yeah, there we go. This Florida one doesn't have 5. Was that got 8? Yeah, so it's not always 5. And that doesn't have to necessarily do with an arm being lost, as they can do and regenerate. We'll see in just a minute. Okay, so they have this radial symmetry. Now, if you remember back when we were talking very early on about the cnidarians, we said they had radial symmetry, and we kind of looked at radial symmetry as being the more plesiomorphic characteristic, right? The more ancestral characteristic was to be radial. That bilateral, with its encephalization, was the way that the other groups went, and that was the groups that we saw. Maybe not more success, but more diversity in. There's more bilateral critters out there than there are radially symmetrical critters. So here we are doing a kind of return back to what we saw before. But that might have more to do with, again, what you said, convergent evolution. It might be something much more advantageous, being where these critters live, all in the ocean, to being this radially symmetrical instead of being bilateral. Now, here's where we throw another trick into it. This is the larvae of all echinoderms. And I know it's kind of hard to look at that. You can see they've labeled some of the parts. It's all very cool. And see the developing water vascular system and all that sort of stuff. But as a larvae, echinoderms are bilateral in body organization. So as adults, these organisms have pentaradial symmetry, or a type of radial symmetry, but as larvae, they're bilateral. All right, now, again, we're bilateral. This is not the only reason that the echinoderms are considered more closely related to us, but I want you to kind of think about that for a second, because in a couple of slides, I'm going to talk about what you might be able to do with a bilaterally symmetrical larval form, all right? Actually, not in a couple of slides, but in the next chapter. But I want you to remember that even though echinoderms have what looks like this round or radial symmetry when they're adults, this is the test. The test is the inside shell of a sea urchin, made of that old stuff, that calcium carbonate. These critters actually have kind of an endoskeleton. They have an epidermis that's covered with these calcium carbonate plates, or these ossicles. So that's what so if you pulled all the spines off of this guy here, which he obviously uses for defense, you would find his test. That's made of calcium, like I said, calcium carbonate. So even though as adult these things tend to have a radial, or even in the sand dollars and the sea biscuit, sand dollar, sea biscuit is thicker, and this critter is way out in left field. We'll talk about those. And these are the sea cucumbers. That knife is there for scale, not to hold it in place. It could, right, Aaliyah? Remember, that's the whole idea behind things looking alike, is do they look alike because of similarity of environment, or do they look alike because of similarity of descent? And, I mean, looking at a group of critters that you've never seen before, you know, you that look alike or look similar, you have to at least entertain both hypotheses to start with. I mean, yeah, a horse and a donkey, you know, they look alike because they're related to each other. But a shark and a porpoise or a dolphin don't, right? So you're right. You say the significant, they have bilateral symmetry like we do, with looking similar, you would think. But remember, it's what you start out as, because you can make, I go back to my example of the chimpanzee infant skull and the human infant skull, right? Very similar when they're younger, very different when they're older. So, yeah, I understand that we look a lot different than an adult echinoderm, many of those echinoderms on the page. But if we keep in mind that the larvae of all echinoderms is bilateral, then we can solve that kind of really switch in symmetry. And that kind of helps us deal with that parsimony problem. If we say, hey, we're closely related to echinoderms, they have radial symmetry and pentaradial symmetry and that sort of ancestral type of symmetry, this idea of, or this discovery, or the significance of them having a bilateral larval form helps us kind of get around. You mean you have to switch the entire symmetry? It's a little less parsimonious. It makes a little more sense, a little more parsimonious if we say, but larvae-wise, they have bilateral symmetry. Okay, keep that on the shelf for just a second, because I think we'll get into chapter 34 today. I can hopefully make that connection about why I'm kind of emphasizing that their larvae are bilateral. What else do we know about echinoderms? They have a nervous system with radial branches, but no centralization of nervous function, so no real brain. Again, it's kind of odd. This is a group of critters that seems to have actually exhibit a lot more of what we would expect the ancestral type of traits. You know, we've seen a brain since our cross-eyed planarian, right? They had kind of a brain, and certainly arthropods have a brain. All right, so as I said, these are the sea stars, the brittle stars. I don't have a picture of a brittle star in here, but we'll show you one later on. The sea urchins, sand dollar, sea biscuits, and this weird group called the sea cucumbers. These are the sea pens, by the way. P-E-N-S, because they look like them old pens that folks used to write, I don't know, whenever they did that. Wasn't it like George Washington guys wrote with a feather or something like that? A quill. Holy cow. I need to go back and do history. Here, uniqueness about the echinoderms. Now, this is only found in echinoderms. This is a water vascular system, and this is and I mentioned to some of you in lab when we were looking at it, this is a skeletal augmentation type of system. In other words, it allows these organisms to move. It allows them organisms to push against something. It allows these organisms to kind of use what we use our skeleton for again. The madreporite, which is that cell, if you look at the aboral, remember the oral is the mouth side, and then the other side is the aboral, which is the top, has a cell that looks a little different than everything else I showed you on the samples there, and that's the madreporite. Through that madreporite, water comes in from the ocean. It's actually a sieve, so it strains out the pieces through this little stone canal, around a ring canal, then out into these radial canals, water. So what we're doing here is we're creating a water pressure system. Now, out on the sides of each of those radial canals are these ampulla. The bulb part of an eyedropper is called an ampulla. So if you look, it does look like an eyedropper. So there's an ampulla, and down below that is a tube foot. All right, now you might have seen these sometimes. You've been at the beach and you flipped over in a sea star and you saw these little tube feet, or maybe what they looked like the way you saw them. If you looked up close, you can see that they have little, they're sort of, well, they're tube feet, but they're flattened at the end. And in this case, you can see what it looks like they're doing. They're sticking to something, and that's exactly what they're able to do. You see, they're able to take water through the madreporite, push it out into those radial canals, and cause it to hydrostatically force those tube feet to elongate, right? So you can see this picture. And then it can reach out, right, and maybe grab a hold of something and then slide, help move itself along, or hold on to something. Or what they use their tube feet for sometimes is preying on bivalves. Sea stars like to prey on bivalves. They like to break open clams and oysters. Well, not break open, I shouldn't say. They like to open them and get in. But, you know, the clams and the bivalves, they got those adductor muscles that keep the shell closed. Sea stars are very patient. They take their tube feet and they wrap them, take their arms with their tube feet, and they wrap it around the bivalve. And they take the tube feet, and when you remove the water from the tube feet, from the ampulla, so when the water flows the other way, you create a suction, don't you? It's kind of like using those suction cups like I was talking about the other day, right? So you pump water into it, then you remove water away. Yeah, that tube foot, which is flattened, creates a suction to it. And that sea star will wrap its arms and its tube feet around that bivalve and just attach it with its tube feet and just gently pull. Just keep a constant force. Well, like any muscle, including those adductor muscles that are inside the bivalve, they're going to get tired after a while, especially if this bivalve can't eat because he's clamped up with a sea star. The sea star doesn't even have to get him all the way open to eat him or her or whatever, right? All he's got to do, that sea star, is get that shell open just enough, because then sea stars can evert their stomach out through their mouth into the mantle cavity of the bivalve with the digestive enzymes, and then just kind of suck that stuff back up. All they got to do is open the shell just a little bit. Hold on one second. So I wonder about that bivalve sitting there waiting. He knows it's coming, right? He's pulling. He knows it's coming. He knows it's coming. And like I said, all I got to do is just crack open that shell a little bit. They can shove their stomach down in between and digest that organism there in the mantle cavity. So they can use these tube feet, like I said, for moving. They can use them for feeding. They can also use them for gas exchange. Any thin enough surface can diffuse and undergo osmosis, just like we've talked about before. All right, so we've got the oral, the aboral surface, the radial arms, the gonads and the digestive glands occur out in the radial arms as well. There are the gonads, that's the reproductive part. These little spines that they have are actual calcium carbonate, and they're protective features. There's the ossicles up here that I was talking about. Those calcium carbonate plates with an epidermis over the top, and that's what gives them, it's kind of what we call it, well, we call it an endoskeleton. Dermobranchia, these little things here are very cool. This allows the sea star to exchange gases with the environment again. So like the tube feet, any highly vascularized surface that's thin enough, you can let diffusion and osmosis. Remember, when you get too big, those things don't work. Down below, the tube feet can also be aided by this adhesive that they use to stick to whatever they're trying to attach to. So they can secrete this adhesive, and then when they need to unattach, they can secrete a disadhesive. I guess that's the opposite of adhesive. Most of these guys are sexual reproducers, but they can split. What you see over here, actually, it depends on the group of echinoderms, but as long as you have part of the central disc, this is the central disc. As long as the piece of the arm, like if this one were to lose an arm in the upper left, but it had at least a piece of the central disc, it actually can regenerate a new sea star. So this isn't a little one growing a big arm. This is an arm that's been lobbed off of another one that's growing another sea star. Males and females, so they're gonorchid, living in the ocean. They're going to release their gametes into the water with their free swimming larvae. And those larvae are going to swim around and disperse and get away from their parents and do all those things that spores did and gametes did out of the bryophytes, and then the seeds and the pollen and everything we've talked about. Whether you're a sea star or any of those other critters, then, you know, dispersal is important. One more last weird thing about this group. In the sea cucumbers, there's a defense mechanism. That's what you're seeing a picture there of a sea cucumber using its defense mechanism. This regeneration of lost parts that we see occurring up here is something that in part, many echinoderms can do to some degree. A sea cucumber, if you mess with it, if you think about it, there are many ways to avoid predation. You could, for example, be like a monarch butterfly. You could make the animal sick. You could run away. You could fight back. You could distract the predator. I think we're talking about the squid. I mentioned squid ink. Many times what that's for, of course, it helps to obscures the view, but it's more of a whoa, you know, just get that predator to flinch for a second. White-tailed deer, if they're in a group and a predator comes after them, you want a young predator, as those white-tailed deer are jumping away from them, those white tails are bouncing, he'll get distracted by all the tails. Oh, chase this one. Oh, wait, chase this one. Oh, wait, chase this one. By that time, they're all gone. An older predator knows, focus on one and just chase that one. So the thing with the sea cucumber, all the stuff you see coming out of there, they've liquefied their internal organs and they then expel them out through the opening. And the whole idea, it's kind of it happened to a friend of mine. We were diving in the Philippines. It's sort of a sticky, messy substance. So the predator hopefully goes, ah! And you know, you're left alone. The fact that they can regenerate those internal organs is what's interesting. The fact that we're embryonically, you know, as adult, we don't look very much like them, but the fact that embryonically we're very similar gives us a lot of interesting ideas of perhaps there is something in the echinoderms that might still exist in some form in us that we can activate and use to kind of regenerate these parts. Is it regeneration or asexual? It's regeneration, because you're not making a whole nother individual. It's more like just replacing. It's kind of like the sea cucumber again. Once you liquefy internal body parts and you make this spewy white stuff, and the predator hopefully leaves you alone, you grow them back. So you're not asexually reproducing, you're just kind of regenerating, yeah. Okay, good. Let's move on now to the different groups of the echinoderms, or the classes. The first one is the obvious one, the Asteroidea. These are the sea stars, you know, ones you're very familiar with. This is a Florida sea star. Remember, once again, the arms don't have to be 5. This is the crown of thorns sea star in California, which causes quite a bit of trouble because it's a voracious predator, and it's destroying a lot of the marine ecosystems. In many ecosystems, the sea stars are actually very important parts of the ecosystems as far as predators. Not the crown of thorns isn't because it's invasive, so it's eating too much. But in many cases, sea stars or members of this group are what we call keystone species. Keystone species, you know, when they built those old archways years ago, that one rock at the top was the keystone. It was usually shaped something like this, right? And that's what held the arch together. They didn't glue it together. You pull the keystone out, the arch falls. In ecology, we talk about a keystone species as the one species that if it was removed or died off in the environment would cause the biggest environmental change. So in the case of the sea stars, even though they're the predators, they keep the marine ecosystem in the balance that it's in at that point. Asteroidea are different than the next group, the brittle stars. Brittle stars we'll see look like asteroidea, but the big difference is that central disk in the asteroidea is not sharply demarcated. What we mean by that is the central disk kind of blends into the arms, or the arms blend into the disk. There's no real obvious round disk. You know, we'll see in the brittle stars in a second that's not the case. The arms have more of an obvious taper to them as well. The sea daisies are really kind of neat. There's only 3 species of sea daisies. This is a sea daisy, less than a centimeter wide, right? There's one species of sea daisy called the Xyloplax genetae, this one here, that was discovered in 1986, just recently. And it was only discovered on the wooden planks, or the wooden, parts of old ships that had sank years ago. Like some of the old sailing ships and stuff. It makes you wonder, where did this critter exist before human beings had wooden ships went out to sea and sank? It depends, Sean. Snakes could be, but again, the keystone species would be the one that causes the biggest change if you were to remove it, or if you were to increase its numbers. It's kind of the one that controls the key to the environment change, or the ecosystem balanced or not. So I wondered, where do these sea daisies live? If now they live on the wooden timbers of old sunken ships. I guess what really us going to sea may have done is help them increase in numbers, right? Because you think about it, trees could fall into the ocean, and that's how sea daisies could have survived. But before we started building wooden things and going out there and sinking them, I wonder if sea daisies were a lot less I was gonna say popular, like high school, a lot less common. Kind of neat. We find new species all the time. It's all very cool. All right, so the next group are the brittle stars, and you'll see right away what I mean by the difference. You notice the central disk. See, it's much more demarcated. It's much more obvious. And the arms don't have the taper that we see in the asteroidea. They're called brittle stars because they're very well known for what we call automatizing their arms. This is one of the weirdest kind of words to say. This is a picture of a brittle star that's undergone automation of its arms. It's another predator duress. It's kind of like the lizard losing its tail, the sea cucumber spitting out its internal organs. It's all that, whoa, get the predator. Maybe the predator will chase the flopping arm for a while, leave me alone. That's what the lizard is thinking, right? Maybe the cat will mess around with this flipping around tail that I disconnect, and I'll run off and be safe. So brittle stars are known for doing that. So that's where hence the name brittle star comes from. They have tube feet, but they don't have suction cups. So their tube feet, if you look down here, have actually ended up becoming more seedy-like. The chitinous material that was on the outside of the earthworms. So it allows it to get traction. These guys are many times crawling along the bottom in the ocean, many times found way deep in the ocean. Matter of fact, the big picture in the back, kind of behind all these other pictures, a bunch of sea stars at the bottom of the ocean. I don't know the depth, but thousands of feet. These guys can actually move around on land too. This might be one, if you've had aquariums, you find that one a lot of times in aquariums. But see this guy? They can actually crawl around on land. So, and again, because of these tube feet that have been turned into these kind of traction devices. Any questions on the sea stars? This next group is really cool too. These are the sand dollars and the sea biscuits, the sea urchins, the Echinoidea. They don't have arms, but they do belay their echinoderm relatedness, because if you look right there, you can see that 5 pentaradial symmetry. And we looked at them on the sand dollars and the sea biscuits. You can see the same thing. They do have tube feet. If we look at the test, this is the test of the sea urchin. The double rows of tube feet are right here. The grayish lines are where the tube feet would stick through. Protected movable spines for defense and locomotion. This group, again, is full of so many surprises and differences. You got this thing called an Aristotle's lantern. See, the sand dollars and sea urchins and all these guys, these are grazers. So they're going to be kind of like the chitin, you know, and the mollusk had the radula to scrape the stuff off the rocks. So the way this thing works, and this is what it looks like from the outside. So if you turn one of these things upside down, these are the mouth parts, these little white things sticking out. But if we look inside, it's something really kind of beautiful if you think about it. It's got a neat name. It's called Aristotle's lantern. And the reason why it's called that is Aristotle, and that little inset you see there, he's the first one to describe it, and he drew a picture of it. So I've taken Aristotle, or they've taken Aristotle's picture, and we've made it a little bit bigger so you can see it down below. Aristotle's, when you crack open a sand dollar and you get that little star thing on the inside, that's what you're getting. You're getting part of Aristotle's lantern out of there. So there's the esophagus coming down, and there's the mouth and the teeth. Look at the way the muscles are arranged. Now, remember I told you before that muscles can only retract. They can't push. So look at the way this thing works. This protractor muscle pulls this way, and that causes this calcareous plate to pull and deform down that way. Now, when that calcareous plate does that, it pushes on the teeth at the top, causing them to open up. I don't want to draw here because I'll mess it up. Now, when the retractor muscle and the protractor muscle relaxes, but when that pulls, the opposite happens, and so the plate then pulls this way, and when you pull this way, the teeth then close. So it's kind of, if I could use my hands, the plate is causing the teeth. As the plate shifts back and forth because of the muscles, the teeth collectively, if you looked at them from the end, are opening and closing. And it's grazing along. But the place you find this is in the echinoderms of this weird group of the Echinodermata. Aristotle's lantern. We talked about Aristotle at the beginning, and we're finishing up talking about him again. He was a brilliant guy. Great artist. Can you imagine having that kind of scientific ability and being that kind of an artist, too? Okay, Leonardo da Vinci, you go, come on, really? You have to be great at everything. I wonder if he sucked at anything. I wonder if Leonardo da Vinci was like someone that you go hang out with, but you like don't eat his food. He's not much of a cook. He seemed to be. This weird group of the Crinoidea that belongs in the echinoderms, these are the sea lilies and the feather stars. This is another example of a living fossil. Again, in this particular case, these organisms don't look like something from the past. They haven't changed much from the past. Remember, I said living fossils have a couple of definitions. They haven't changed much since the Cambrian explosion. Organisms in the ocean oftentimes don't, especially if you live in areas of the ocean that don't change a lot themselves. Remember, it's the environment that brings organisms to change. In the middle there, you can actually see a fossil of some early Crinoidean echinoderms from back during this era. Males and females with free-swimming larvae, a sessile juvenile that then turns into a sessile adult. Although the feather stars are free-moving, look down below. That's a feather star. It's a feather star crawling up on a beach. Can you imagine landing on the beach, kick back on, just getting some sun, drinking a beer, and watching this thing crawling out at you? What would you think right away? It's a weird-looking thing, man. I think that this weird giant spider's coming after me, but they can actually move up on their arms and crawl out on the beach. And since they have those dermal branchia that I showed you before, in many cases, they can use those to actually do some gas exchange. But most of them kind of look like the sea pens that you see there, the sea lily sea pens. They have a mouth that faces upward with a U-shaped gut, putting the anus right next to the mouth. And it's kind of like the gastropods again, with their torsion thing. Kind of have to play with the hands that dealt you. That is a weird-looking thing, man. And then finally this group, the sea cucumbers, which, by the way, are edible. Often they are consumed. Sea cucumbers are called that because they look like sea cucumbers. Very soft with not much of an endoskeleton. The oral tube feet, the ones up around the mouth, have actually been modified into feeding tentacles. Reach out and grab their prey. We've seen that a million times with different kinds of ways. You can do it with cilia. You can do it with the Lophophore mechanism that I was talking about, the Lophotrochosaura. These are important detritivores in many cases. Okay, detritivores, organisms that feed upon detritus, decaying organic matter. They're going to be recyclers. Breathe through their anus. What do you mean by that? I don't know, Sean. I don't know about that. I know you can eat them, but I don't know about. I don't know. Look at the thing on the plate. I guess I don't know if you sliced it off. I don't know. I'd try it, I guess. I guess I always kind of feel like I'd try something. Except the sharp stick in the eye, but I might try them. Yeah. That'd be cool. Find some place where we can eat them. So they have this thing called a respiratory tree in their anus. That's how they're able to breathe through their anus. And a respiratory tree is basically, if you go up, it's a bunch of like very thin passageways that water can diffuse into, or water can flow into, and then it's thin enough. A lot of surface area, so you get a lot of diffusion going on there. Yeah, it might. Okay, so that finishes up our nearest ancestor, and for the next one, I'm going to go to the next chapter. So I would like to start that. So let's get on chapter 34, if you don't mind. Let me switch over. Yeah, but is it? Because it's like pufferfish, right? You can eat pufferfish, but you got to be careful the way it's prepared. If I remember right. These slides to load. Sorry, folks, I didn't have the slides loaded. But yeah, I think that it's like with a lot of species, you just got to be careful with how you eat them. While we're sitting here waiting this to upload, I'm sorry I didn't have it pre-uploaded. I thought I had, but if you have any questions. Course evaluations come out tomorrow. You'll be getting emails. As I said, please, I really, really need you guys to fill those out. Even if you don't feel like going through all the yes, no, number type of questions, comments, folks. Give me comments. I'd like to hear about. I mean, I would like you to fill out the whole thing. But I do really want to hear about things you liked about the class, things you didn't like, things you liked about the book, anything, lab, anything. That's the only way I know how to modify my course and, make it better. If you guys tell me what's working and what's not working. Lab assignments that you liked, ones you didn't like, just, anything. All right, this is apparently I'm on dial-up today since it's taking forever to load. Thank you. I appreciate that, Sean. You can write good things on there, too. I mean, if you want to, thank you, Leah. If you want to, that's great. I like hearing good things about myself. But I would also, if you had things that you thought worked better than other things or things that didn't work so well or anything like that, I would like to hear about them. Because that's the whole field notebook thing that I have you guys do sprang out of a comment that I got in a survey years ago when I started doing them. I had only done them the first semester. And a student raved in the comments about how wonderful it was and how it got him out of into the woods, and he got to appreciate Florida a lot more than he ever did before and all this stuff. And I don't know. I just said, well, then this is a good assignment. Why is 34 taking so long? Well, we got plenty of time. That's what I'm hoping, especially living in Florida. There's so many unique things around here. It's converting. So what do we got next week? Monday, and then there's no lab. I didn't see that chat that just came through. I was on the other screen. Yep, that's right. And that's the problem, too. And again, remember, there's a difference between exotics and invasives. Exotics are there, not causing a problem. Invasives, There we go. Chapter 34. Thank you. Finally, invasives are there and causing troubles. So let me share this with you. Chapter 34. Okay. Really? What, from where? I mean, where was... Oh my goodness, where are you? Yeah. Those things come over on boats and ornamental plants and all kinds. There we go. What? I want to get at least into, there we go, into the phylogeny. Stay right there. Don't do anything. Origin and the evolution of the vertebrates. Okay, so this is a fossil of a, this is probably a placoderm. We'll talk about more about placoderms later on, but that's an ancient fish. They're not around anymore. Unfortunately, they were very cool looking fish. They were called placoderms because they had these plates over their skin, because back during this time, when this evolution of the vertebrates really started to take off, or animals in general, really, there's a big arms race going on. There's a lot of things eating each other. Nothing drives evolution, as I've said before, like the way of trying to protect yourself from being eaten, as well as trying to find things to eat. Okay, so the echinoderms up here at the top is where we were. About 57,000 species of vertebrates. That's not a lot, right? Remember, there were 250,000 species of flowering plants. So 57,000 species of vertebrates. This is tend to be the more synapomorphic or derived groups. So the diversity many times in organisms that tend to be, let's say, more complex tends to be a little less. So you might not be surprised by 57,000 species only. What you might be surprised about, though, is, again, this idea that the Aristotle and his ladder leading one and the other is not really the way it happened. The first vertebrates appeared back in the Cambrian explosion, about 550 or so million years ago. Back during that time, before and during the Cambrian explosion, there were already vertebrates. So it wasn't, it's like once we got out of the protozoan group, all these different groups of organisms kind of came out of that group relatively simultaneously, if you let me say a few million years of simultaneously. Fish were the very first ones. And initially they didn't have jaws, and that worked well until somebody came up with jaws. And having jaws in an environment where everybody else is jawless is like bringing a bazooka to a knife fight. You know what I mean? You got the advantage, the big advantage. Now, what I mean by jaws is things that you can eat, that you can grab, that you can chew. And Sean, you're very familiar with this, of course, with your snakes. Jaws are extremely important to organisms, and you can imagine in that in that early ocean, as everybody's swimming around trying to stay alive, if somebody figured out a way to crunch and grab you with their mouth, oh my goodness, you're going to have to do something to survive. So the amphibians came along, and they came out of a fish-like ancestor, and they moved out onto land for a variety of reasons. It might have been to escape some of the predation. Amphibians had jaws too, so it wasn't like they didn't have jaws, but maybe escape some predation pressure, and if for no other reason than, holy cow, there's nothing else out there on land, right? Even if we can move on the land a little bit, even if we got to come back to water to reproduce, we can get out on the land, and we can exploit some of these environments that the fish can't. See, if you look at this big group here, we're going to go through each of these. The cephalochordates, that's the Amphioxus in this group, but these are all from here up. These are all pretty much going to be bound, in the most case, you're going to be bound to the ocean, right? The fishes, the different types of fishes. So when you be able to move out onto land, when you be able to have these limbs with digits, see those nodes, those characteristics that arose there, then you really would have had an advantage, right? We still got to go back to water to lay land. If you became a reptile, though, when the evolution of the reptiles came along, then you became a dominant land predator because you could reproduce on land, right? It's about the egg. If you can lay your egg in water, you got to live near water. But if you can lay your eggs out of water, like the reptiles figured out how to do the reptiles that we now. That's why. But if you look at it, where are the birds? Well, remember now, the prevailing thought is birds are reptiles. We used to teach this as there were the fish, the amphibians, the reptiles, the birds, and the mammals, but that's polyphyletic, folks, saying it that way. Birds are reptiles, so they belong in that reptilian group. That's why you don't see them. All with those amniotic eggs. That's the big thing. Having an amniotic egg allows you to lay your eggs away from the water because the amnion is a sac of fluid around the fetus that keeps it from drying out. Which ones are you talking about, Sean? I'm sorry. I see your question, but I don't remember exactly when that came in. The young being in different environments than the adults. Oh yeah, absolutely. Resource partitioning. Yep, that's kind of like with the insects, the butterflies and the pupa and all that. Yep, absolutely. The birds and the mammals. Yeah, the birds and the mammals benefited from the asteroid strike because they became dominant after the Cretaceous mass extinction. The dinosaurs were doing quite well as reptiles on Earth, running the Earth for the 200 million years they had, but could not keep up with that quick change that was brought about by that asteroid. So they went the way of extinction as well. That left open an environment for the rest of us, for the birds and the mammals. So, all right, let's talk about a chordate. What makes a chordate a chordate? Like I said, it comes from the Latin word that means string, so it's obviously talking about a spinal column. Now, here's the thing where we talk about the technicalities of putting organisms and groups together, and has a lot to do with similarity of embryonic development. As I said, the nearest relative of all the chordates is the echinoderms. For one thing, we're coelomates like they are. We're deuterostomes, and again, there's that bilateral larvae that I talked about. But before we get to that, let's look at all chordates have these four features, and I put in italics to remind you, at least at some time during their life. To be a member of the group Chordata, to be a member of the phylum Chordata, you have to have had these four bolded characteristics you see listed there, at least during some type or time of your life. Okay? Dorsal nerve cord. Remember, that was very different than we saw with the earthworm. They had a ventral nerve cord. That's unique to vertebrates. So you have to have a dorsal nerve cord. You have to have a notochord. Now, we don't have a notochord anymore right now. As a matter of fact, our notochord has been replaced to parts of the vertebral column and the discs that are in between the vertebral column in humans. That's what's left of our notochord. Now, what is a notochord? A notochord is a stiffening rod. So if you look down below, this is the amphioxus, the little model of the critter. Notice the notochord. The notochord, the brown thing, is the string, but it's not made of bone. It's just a stiffened rod. And of course, the stiffening in the rod allows these organisms to get bigger and have more, and allows them to, to get bigger. They allow them to get bigger and allow them to get more diverse because they have this skeletal type system, okay? At some time, we all had pharyngeal or gill slits. If you look at this kind of cool picture here, we're looking at a fish and a salamander, tortoise, chicken, rabbit, and a man. If we go back during a similarity in embryonic development, you find at one time we all look very much alike. And we all have gill slits. Now, if you're a fish, those gill slits are going to become your gills. Salamanders and many amphibians have gills at some time in their life as well. Our gill slits become the structures of our throat. They don't become our lungs, but they become the structures of our throat. The nerves, the musculature, and that little thing called eustachian tube is the remnants of your gill slits. Gill slits, pharyngeal slits, or pharyngeal pouches. Down here on the right, chicken and the human embryo, pharyngeal pouches. This is the human embryo. At some time in your life, all of us had a post-anal tail. Ours has been reduced to just a couple of bones, but in some cases, obviously, if you're a chordate, you're known for having your tails, right? Chordates are also possess these things called somites, and I've indicated the somites. And one real cool thing about the somites is you can see the segmentation in them. The somites are the little white lines that you see in the picture there, and the divisions you see in the chicken. Somites give rise to things like the axial skeleton, so the spinal column part of the skeleton, tendons, muscles, and again, it's evidence of the benefits of segmentation. Remember, I told you before that segmentation doesn't necessarily mean you're related, but it is a trait that many organisms picked up on that proved to be advantageous. Because when you're segmented, you can specialize. Remember, the insects really went crazy with that. So who are we going to talk about in the chordates? Well, obviously, we're going to talk about the critters you're most familiar with, the fish and the amphibians and reptiles and the birds and the mammals and those guys. But we're going to start with some chordates that you might not be quite as familiar with, the tunicates and the lanceolates. But before I get to them, I want to talk a little bit more about this echinoderm bilateral symmetry thing. I think this will probably be the last thing I talk about today, too. So up in the upper left again, I remind you that's an echinoderm larvae, and they're bilateral, okay? Now, if you look down below, right below, it looks like a sperm cell, but that's actually not a sperm cell. That's actually the tadpole larvae of an organism known as a tunicate or an ascidian. Now, ascidians exist today. They are chordates. This is a picture of ascidians. Really kind of cool. They're filter feeders. Many colors. They're called tunicates because they're known for a sac around them, a tunic, all right? Now, if you look in the reproductive life cycle of the adult ascidian, when it reproduces, sperm and egg, out into the water, fertilization, when it reproduces, it forms first a tadpole larvae with bilateral symmetry. That tadpole larvae then disperses and eventually settles down as a sessile adult. That's the way ascidians do it today. This is a picture of an ascidian or a tunicate, which is a type of chordate, larvae. This is a picture of one, bilateral symmetry. Here's where we think the connection occurs, all right? See where the question mark is? If we jump ahead in time, this is kind of like trying to fit the pieces in from both ends. We go back to our buddy, the astracoderm. There's the, I said placoderm earlier, the astracoderm. Astracoderms and placoderms were two fishes around this time, very similar to one another. But some of the earliest fish out there were the astracoderms. Okay, and the astracoderms, you see the picture there, were very fish-like looking, kind of armor-plated, known more for jaws than anything else. Placoderms were really ones with the heavy armor plates because when jaws came about, then it was important to protect yourself. But if you notice that astracoderm, and down below, could reasonably have evolved out of some sort of vertebrate ancestor that underwent this thing called a paedomorphic, right? It was a paedomorphic vertebrate ancestor. Paedomorphic fits into something called Garstang's hypothesis, up above known as progenesis. Here's what do we mean by that. If something happens during development of the larvae, whereby the larvae becomes sexually mature while it still looks like a larvae, then from that point you could evolve another lineage of organisms. So here's what we think happened. Some bilateral echinoderm larvae or an ancestor of this bilateral echinoderm larvae had some sort of a tadpole-like larvae that was bilateral. And during little changes that occurred very on in its development, this tadpole larvae was no longer a larvae. It was an adult organism that looked like a tadpole. That's what we mean by the development of sexual maturity in a larval form. And that larval tadpole form, no longer a larvae, could look an awful lot like early fish, like some of the astracoderms. Does that make sense what I'm talking about? So what we did is evolution, and these mutations occurred in the larval form. So I'm talking about the jump where the question mark is, that ended up creating a vertebrate ancestor that actually looked like the larvae but became sexually mature, so became the adult itself. That's what we mean by a paedomorphic vertebrate ancestor. A paedomorphic organism is an organism that looks like the larval form but can reproduce itself. Now, this is, in a broad sense, showing us how fish may have got their form. All right, so the vertebrates, the first real group of vertebrates were the fish, and we think maybe it came about because of some mutation that occurred very early on in the embryonic development of these larvae that we know exist today. So, so some ancestral Ascidian larvae that would have evolved from or ancestral citizen larvae like the larvae, the tadpole one there, that would evolve from some bilateral ancestors that gave rise to the echinoderms and larvae, and the echinoderms because their larvae is bilateral. Tadpole larvae is bilateral. The connection is if you took that tadpole larvae and it became sexually mature in that shape, then you have the beginning of fish. I know I kind of went over that a million times. I think I went around my elbow to get to my thumb, but did I confuse you, or do you understand what I mean by Garsting's hypothesis? Garsting's hypothesis explains what might have happened here. Yeah? How about you, Leah? Are we good? Yeah, so again, what we're saying is these adult Ascidians still have these tadpole larvae. So sometime in the past, maybe one of these old ancestral adult Ascidians, its tadpole larvae, went through a mutation and became the beginning of fish. Yeah. There's a lot of good, neat videos online about Garsting's hypothesis too. Take a look at those, and you can find something that might help to get a better visualization. The problem with understanding, I think, is the big jump I'm making. We're going from tadpole larvae to fish. Yes, there you go, Sean. That's actually better than I said it. Aaliyah, what Sean just said is right. The tadpole larvae began able to reproduce, so it wasn't, by definition, a larvae anymore. It was the adult. There you go. Thank you, Sean. Much better than me. Thank you. All right, good deal. Let me turn the recorder off there.